Hi, hello. Thank you very much for inviting me to speak on this year's BLS AGM. I very well remember last year when we all met in Edinburgh and how nice it was. I will do my best to give a good performance in this online meeting. Thank you very much for the invitation. I will talk about lichen stress eco-physiology today, asking the question how lichens can respond to a changing climate. Well, what is it that makes a lichen a good indicator for environmental stress? According to Grimes' CSR, or the Universal Adaptive Strategy Theory, lichens belong to a grouping of organisms displaying traits of stress tolerators. In comparison to other organisms, they show high resistance to environmental stresses, along with slow growth rates, high rates of nutrient retention, and low phenotypic plasticity. Lichens do equilibrate their internal water content with the surrounding atmosphere. They are only active when they are wet and hydrated. As a consequence, their growth rate directly translates to the precipitation and water availability regime of the habitat. So this means if we are to study effects of climate change on terrestrial vegetation, lichens can serve as sentinels of that change. And this ability mainly derives from lichens being a symbiotic life form. In the Lycan Symbiosis, each partner is playing its role. While the photobiont photosynthesizes and transforms inorganic CO2 into organic building blocks, the microbiont provides a moist environment and further respiratory CO2. The ratio of how many photobionts are hosted in a lichen thallus is not set into stone. We know that lichens can meticulously adjust their consumer to producer ratio. So this is the ratio between the myco and the photobiont. And they can adjust this according to the temperature regime of the habitat or even to the nutrient availability. In a warmer environment, more photobionts relative to the fungal mass are needed to sustain a lichen thallus. As such, this phenotypic plasticity, so the change in the organism's morphology and the physiology in response to that, are one way for a lichen to acclimatize to different environmental change. Another way of responding to different environmental conditions is photobiont switching. Photobiont switching is the mechanism which allows a specific lichen fungus to associate with new algal partners. And this has been shown to occur throughout lichen evolution. Being able to switch a photobiont actively would allow lichens to acclimate to changing environmental conditions, presumably by selecting an algal partner that's specifically adapted to those environmental conditions. Both mechanisms, the phenotypic plasticity and the photobiont switching, would allow a lichen species to withstand pressure from environmental and climate change. And in this presentation, I want to explore these two mechanisms for a common soil crust lichen, Psora decipiens. Psora decipiens is a pale scrumulose crustose lichen, conspicuous, white, pruinose, often upturned margins. The lower cortex is white, very poorly developed or absent, and the attached hyphal net penetrates the substratum. It is a cosmopolitan lichen, and it is well known as an indicator species for intact biological soil crusts in climate stages. We have found this lichen as a common representative in biological soil crusts in the high alpine regions of the Großglockner in Austria, which you can see in the upper picture here, as well as the driest regions of Europe, such as the Almeria badlands in southern Spain. You can see on the lower picture here. Looking for these lichens is a wonderful task, given the amazing landscape, but also quite exhausting. As you can see here, this is me after an exciting day lichenizing in the high alpine. Well, the finding of the lichens in these two very distinct sites was initially quite astonishing to us. Here you can see the differences in the climatic conditions between the two sites. In the high alpine, Großglockner in Austria, and the southern Spanish side, also called Tabernas. However, these graphs are quite tricky, so I will show you some overall climate data in a table. This is a table contrasting major climate data for these two sites. Most importantly for you to look at is the mean temperature here, which is only just above 0 over summer in Austria at the high alpine site, but almost 23 degrees over summer in the southern Spanish side. Also interesting to recognize is the difference in relative humidity. At the site in Austria, this is above 90% for both summer and winter, but only between 50 and 60% at the site in southern Spain. An average of 558 millimeters of precipitation fall in the high alpine, and only 25 millimeters of rain fall at the Spanish site in Almeria. So we can really say that these two sites are indeed climatically distinct, and it is surprising to find a lichen of the same species at these two sites. This is the ideal setup for testing the hypothesis on lichen plasticity being triggered by external climatic factors. So the first thing we did was to compare the morphology of this lichen and also the physiology of the two populations. This work was published in the Lichenologist a couple of years ago. On the top, you can see an image of a cross-section of Psora decipiens thallus coming from southern Spain. And on the bottom, you can see the same picture, but taken from a specimen collected at the high alpine site in Hochtor. And you can see that there are indeed significant differences. In fact, we were able to quantify these differences, and we found the most significant difference morphology-wise indeed in the photobiont to microbiont ratio. This is here expressed as the ratio between the photobiont layer thickness and the medulla layer thickness. And this was twice as high for the population collected in southern Spain, supporting that the hypothesis that there is significant phenotypic plasticity in the lichen consumer to producer ratio in response to the regional climate. But what would that mean for the lichen physiology? What does the morphological change respond to changes in lichen performance and growth as well? So we've been looking into physiological traits, such as how much water can the lichen take up, how quickly can the lichen take up the water, and how long can it hold on to it. And everything we found was that the lichen from the site in southern Spain had significantly altered its morphology so that it could make the best out of the little water available on site. One such adjustment, and that's the adjustment I want to highlight here, is the cracked epinegral layer, which we suggest acts like a funneling system to direct the water quickly to the photobiont layer. And this is something that we didn't see at all in the alpine specimens. Another interesting experiment was to look at the different modes of water uptake in this lichen. In this experiment, we had glued a single lichen thallus to a very fine needle and then submerged the rhizins very carefully into the water, which was colored in ink. And then we took time-lapse images that would allow us to detect the location of the blue water. And we found the blue water to be in the photobiont layer already after 17 seconds. And you can see it on this picture here, slight green bluish layer just above the medulla. We could also show that the water, once it is in the photobiont layer, it remains in the photobiont layer. It does not go into the medulla. And you can see this on this final picture there. After 2.5 hours, the medulla still being more or less white and only the photobiont layer is blue. So with these results, we knew that there was a great plasticity within this species. Plasticity indeed to such an extent that we could show different physiologies for the two distinct populations. And then given these acclimations are most likely the results of long-term acclimation to the regional climate conditions, the question arose how flexible the lichen would be in response to more acute changes, such as we predict them from climate change scenarios. So the working hypothesis behind this would then be not to wait for morphological changes to materialize, but to switch photobionts quickly in order to acclimate to the local climate. And we tested this in a large-scale transplantation experiment. Again, Psora decipiens is the model organism here, and we used it to transplant it between sites within Europe that had well-established biological soil crusts. So we transplanted patches of Psora decipiens between sites in southern Sweden on the island of Öland, a site in the middle of Germany at a small town called Gössenheim. And then the two sites you already know, the high alpine site in the Großglockner Hochtor and the southern Spanish site in Almeria. And we fixed the transplants with fishing line onto the ground, and you can see the remainers of the fishing line on those pictures here. And we left them at sites for a full year to grow. The transplant experiment as such was successful. You can see this in the lower table. This is where we show how many lichens showed new growth after the full year at the new site. So the first thing we did with this experiment was to look at the evolutionary relationship between the different populations. What you see here is a phylogenetic or evolutionary tree, basically a branching diagram showing the evolutionary relationships among the different populations of Psora decipiens, based upon similarities and differences in their genetic characteristics. And we could show that Psora decipiens is indeed composed of at least 4 genetically and geographically distinct groups. The German and Swedish sites are sharing a genotype, and this highlights similarities in the climate as well, and lends support to the suggestion that the genotypic and morphophysiological variations found in this lichen have a climatic determinant. The population both in Spain and Austria were distinct. But with regards to the photobiont switching hypothesis, what is even more interesting in this transplant study was whether or not the photobionts survived the transplantation. And what you can see here is a table showing in which transplant scenario the photobiont layer remained alive. This is indicated by a blue tick, and where it vanished, this is a red cross. So transplant and photobiont survival only occurred in sites that had similar or milder climatic conditions than the original. The lichens surviving transplantation between Sweden and Germany is unsurprising, considering that they are very similar in their environmental conditions, and they have a corresponding microbiome genotype. Therefore, the transplants did not necessarily need to acclimate any further than what would have already occurred on a seasonal basis in the native habitat. Also, the transplantation survival from an extreme to a mild site, for example, from Spain to Sweden or Germany, can be explained by climatic conditions. An extreme site is not extreme all year round, and conditions at certain times would be very similar to those in a mild site, during which the lichens would be active, compared to harsh conditions, for example, the summer in Spain. The most striking finding here is that any transplantation effort to the extreme site in Spain has been unsuccessful, meaning that photobionts had not survived the extreme shift in climatic conditions. So what was going on? We found that indeed the lichen grew initially, new growth was found, but if we looked at the cross-section of these thalli, we found there was no longer a fit algal layer. And you can see some examples of how that looked like on the pictures here. The transplants were installed in October, so during the growing season, 6 or 7 months before the harsh summer returned. Consequently, it can be assumed that there was sufficient time for new growth before the climatic conditions became detrimental to the transplanted lichen. In any case, we could not show any signs for this lichen to take up a new photobiont strain, and this would be consistent with current knowledge about photobiont switching. It seems unlikely, although not impossible, that a fully formed thallus would be able to integrate new algae. So what does that all mean? What can we learn from this? Well, our experiments have shown that even a very plastic lichen with a high phenotypic plasticity, such as pseudoidicipiens, might have problems with adjusting to new environmental conditions if they are abrupt and extreme. So is this relevant at all? Are such conditions to be expected? Well, in recent decades, especially since 2003, Europe has experienced an exceptional number of pronounced heatwave events. And in the light of this research, our lichen communities might actually be affected in their performance and survival changes by such events. I hope you found this interesting. Thank you very much for listening. Please let me know if you have any questions. Bye.